Hi, everyone. Welcome to today's Lunch and Learn webinar on the Ovation Research Network and the Network Participation Agreement. I'm Barry. I'm the co-founder and CEO at Ovation, and I'm so glad you're able to join us today. We're looking forward to sharing this exciting opportunity with you and to answering your questions about the Network Participation Agreement. Some of the folks on the call may not be familiar with Ovation, so let me just give you a brief introduction. We're a technology company. We started in the healthcare space in 2017, and we're backed by some of the leading life science venture firms and technology investors. My co-founder and I came from scientific research and life science backgrounds, but we saw an opportunity in the clinical space to shorten the distance between clinical data and research, specifically when it comes to making precision diagnostics more accessible. Today, we build lab software that helps independent molecular diagnostic labs run their business through our LIMS offering. But more importantly, we build the Ovation Research Network, which is what we're here to talk about today. A network participation agreement governs labs' participation in the Ovation Research Network. So today we're going to cover important aspects of this agreement. Our goal is to orient you to important sections of the network participation agreement and to answer common questions about network participation along the way. So here's what we'll cover. First, we'll talk about how your lab is compensated for participation in the research network. We'll discuss expectations regarding specimens and information provided by your lab to Ovation. We'll cover legal details and technical resources for de-identification of patient information and how Ovation ensures patient privacy. We'll talk about how your lab should think about consent and patient authorization strategy when participating in the Ovation Research Network. And finally, we'll talk about eligibility for providing follow-on research testing services to the Ovation Research Network and the network services addendum that describes those services. Some housekeeping details. This recording will be available on demand sometime in the next 24 hours. Please ask questions. Today is for you to make sure that you understand the network participation agreement. You can put your questions in chat or in the Q&A function in the Zoom webinar. We'll make sure we answer all of your questions, either live today or by follow-up afterwards if we run out of time. Okay, so first up, maybe the most important part of this agreement is understanding how your lab is compensated and reimbursed for participation in the Ovation Research Network. Of course, we recommend that you read the terms of Section 2 about compensation carefully within the agreement itself. They'll be specific to your lab, but here we'll give you an overview of what they describe. There are two main ways your lab gets paid as part of as a participant in the Ovation Research Network. The first is reimbursement. We'll reimburse your lab for the reasonable costs required for you to prepare and transfer remainder patient specimens and associated information to Ovation. You'll work with our clinical affairs team to determine what those expected costs are on a monthly basis, and you'll send us an itemized invoice for those costs at the end of each month. We'll pay you within 30 days of receipt of that invoice via an ACH transfer. The second way you're paid as part of the research network is through compensation. We'll compensate your lab each time we use a specimen in a research project that when that specimen originated from your lab. And we can compensate your lab for providing services to the research network under a network services addendum. We'll cover that later in this presentation. When it comes to reimbursement, there are a couple of core things to note. First, as I mentioned, your lab will work with our clinical affairs team to determine the costs associated with preparing and sending samples from your lab to the biobank. The amount that is determined in that consultation will be referenced in the network participation agreement. If your lab requires reimbursement for an amount that's beyond that baseline specified in the network participation agreement, you'll just need to seek prior approval from Ovation before submitting your invoice so that we know it's coming. On the compensation side, your lab can earn net new revenue in two ways. The first time a sample is used, a sample from your lab is used as part of a research project, your lab will be compensated based on the type of sample used. Depending on what type of specimens your lab is sending to Ovation, that fee schedule will be determined with our clinical affairs team and will be outlined in Section 2 of the network participation agreement. The other way your lab can earn money is through research-related follow-on testing. Now, these options are mutually exclusive. If your lab is eligible for follow-on testing and we send samples to you for that project, we'll pay you for doing the research testing. We won't pay you separately for usage of the sample. If your lab has the capabilities to perform the required research testing, we'll sign a network services addendum with you for that research opportunity. And that addendum will specify the rate and schedule for your payments for that research service. The next important section of the network participation agreement is provision of specimens and data. This is Section 3, Part B. In this section of the presentation, we'll outline some of our expectations regarding patient specimens and information that your lab will provide to us as part of your participation in the research network. There really are two core expectations. The first is that your lab will provide excess or leftover patient specimens and associated information to us to be stored in a biorepository, unless otherwise instructed. Your lab will be responsible for packaging and shipping these specimens and the associated information, and you must do this in accordance with applicable laws and any written instructions from us. Now, while it's your lab's legal responsibility to ensure the coordination and transfer of these specimens and information follow applicable laws, especially HIPAA, you'll execute a business associate agreement with us. That business associate agreement is included as Exhibit C to the network participation agreement. And by executing that BAA, it will allow Ovation, as your business associate, to assist you in performing those activities. So our experts will help your lab adopt a process that is amenable to your current operations, making sure that you're meeting the highest standards of both specimen quality and regulatory requirements. So some common questions that come up when reviewing this section. First, what type of specimens are useful in the research network? Generally speaking, we're interested in most types of specimens that your lab collects, as long as they contain enough human DNA or RNA to extract value. The model of the research network is to cast a wide net and capture specimens from a broad patient population across many therapeutic areas. The specimen types that your labs will send to our biorepository will be outlined in Section 2 of the agreement, and that list will be developed in consultation with our clinical affairs team. What are the minimum requirements for a specimen to be sent to the biorepository? Here are the requirements. First, the specimen must have minimum patient information attached to it so that it can be properly de-identified. That includes full name, date of birth, sex, and a valid zip code. We'll talk about the de-identification procedure and process later in this presentation. Second, the specimen quality must be sufficient that integrity of the nucleic acids has not been compromised. And finally, the specimen must have the proper entitlements documented for research testing. We'll talk about those specifically in the consent and authorization section of this presentation in a minute. So to review, sufficient patient information, sample integrity, and research entitlements. Some labs ask if they can biobank samples in their own facility and still participate in the Ovation Research Network. The answer is definitely yes. We understand that for regulatory reasons, you may not be able to move all of your specimens off-site. And in that case, your lab will index those specimens using our software that we'll provide and you may continue to store them on-site. You may also choose to serve as one of the distributed biorepositories for the Ovation Research Network, and we can pay you to store specimens that we own at your location. We do that using a network services addendum. Once we request a specimen for a research project, if you've been keeping them on site, you'll determine which index specimens have sufficient material for research use as part of the verification process. And you'll send those verified specimens then on to the Ovation biorepository to perform additional research testing. Operationally, how often will your lab send specimens to the Ovation biorepository? Well, the shipping process will be custom built based on the specimen type, your current on-site storage capabilities, and what works best for your lab. After sending clinical test results to patients, you're welcome to keep those remainder specimens on site based on the cadence that makes sense for you. Typically, we see labs sending us a batch of samples every week, but we're happy to work with you to set up a more or less frequent cadence to fit your lab operations. Typically, the specimens included in the batch will have provided patient results in the prior 3 to 10 days. But again, this depends on specimen type and regulatory constraints of the particular test being performed. So now let's step to the next section and talk about the legal and operational details of how we de-identify patient information and how we ensure patient privacy throughout its lifetime in the Ovation Research Network. And let's start by aligning on the big picture, the promise of precision medicine and a challenge that healthcare has in bringing that promise to patients. Precision medicine offers the promise of life-changing therapies. These precision therapies could be, that are selected based on patients' personal genotype and phenotype, offer improved outcomes, reduce complications, and in some cases, dramatically extend survival and quality of life. Molecular diagnostic labs have a foundational role to play in the delivery of precision therapies, but you may have noticed that not all patients who could benefit from precision therapies are getting them. It's critical that together we protect the privacy of individual patients while maintaining the ability to derive value and conduct research that will benefit the general population. We want to ensure that you understand what happens to leftover patient specimens and information, including your lab's legal responsibilities regarding de-identification, the steps we're taking to ensure patient privacy is protected, and the possibility that identifiable materials may enter into our partnership in the future. All these activities are outlined in Section 3 of the Network Participation Agreement. And in that section, you'll note that it's your lab's responsibility for ensuring there's a process in place to de-identify patient specimens and information in accordance with federal requirements. Well, these requirements state that your lab is ultimately responsible. The Health Insurance Portability and Accountability Act, or HIPAA, permits a business associate to de-identify patient information on behalf of a covered entity, such as your lab. That's why we're asking your lab to execute a business associate agreement, again, Exhibit C of the NPA with us. That BAA allows us to handle the de-identification process on your behalf of information before it's stored in our database and to de-identify patient specimens before they're stored in our biorepository. By managing this de-identification process, we're able to bring these samples and the data into the research domain and to drive value and acceleration of life science research that ultimately brings the promise of those precision therapies back to patients and to the community. That's why we're here working on this together. So, patient information starts as identified patient information in your lab. This is for its clinical purpose for diagnostic use. Patient information is then de-identified using advanced technology that replaces personal identifiable information with a one-way Ovation-specific cryptographic hash. This creates a fully identified patient record in compliance with HIPAA. That cryptographic hash protects patients' personal identifiable information, as well as any associated clinical information from your lab. And once that cryptographic hash is applied, technical, security, and administrative controls ensure that it is not possible to retrieve the patient's identifying information or to reassociate that patient's identity with the de-identified data that's stored in our database. This process transfers patient identifiable information to de-identified research information. The patient specimen is also de-identified upon receipt at our biorepository. As shown in the middle of this figure, the sample ID and any PHI on the specimen's label is replaced with the same cryptographic hash, a global ID for our biobank. Genomic information that is generated from research testing of those de-identified specimens, for example, whole genome sequencing results, will be associated only with that cryptographic hash, not with the patient's identifiable information. So the cryptographic hash allows individuals' records in our data set to be linked with a record for the same individual in a different data set without ever exposing PHI of that individual. So internationally recognized experts on health data privacy have assessed Ovation's database, its design and structure, our policies, and our data processing routines, and determined that they safely de-identify patient materials. The Ovation system and the Ovation Research Network thus satisfy the expert determination method outlined in the HIPAA privacy rule. Now, I want to emphasize Ovation today in the Ovation Research Network is only using de-identified patient specimens at this time. However, there are two important points in Section 3, Part D that explains how identifiable patient information may enter into our partnership together in the future, and I want to call those out. With additional patient consent, we may look to collect identifiable information from patients who enroll in clinical trials as part of the Ovation Research Network. With an amended network participation agreement and revised patient consent, we may also look to collect identifiable information from patients during regular clinical testing. Now, this identifiable information is valuable to researchers who may want to contact patients for participation in clinical trials. And therefore, it's also valuable to patients who want to be contacted so that they can find clinical trials specific to their therapeutic need. We're not doing this today, but in the spirit of transparency, we want to make you aware of our desire to enable this collection of identified patient material. Doing so would require an amendment to the network participation agreement. We don't want there to be any surprise if we re-engage with you on this matter in the future. But I want to emphasize that by signing the network participation agreement today, your lab is not making any commitment with regard to the collection of identified patient materials, except to have the discussion with us in the future. So, some common questions about the de-identification process. Labs often tell us that they would prefer to de-identify specimens themselves before sending that information or those specimens to Ovation. We can build that into your lab's process so that you can perform de-identification of both data and specimens before sending them to us. We'll provide you with the technology and tools to accomplish this process as part of your normal workflow if you desire. Who will have access to information in the Ovation database? Well, access to our database of de-identified data is limited to members of the Ovation workforce who need it. And the de-identified data will only be provided to researchers with approved study proposals. The data sets that we provide to those researchers will be limited to what they need to access in order to conduct their studies. And our data license to those researchers includes legal provisions requiring them to protect the security and privacy of the data set. Our database is built, as I mentioned, with technical, security, and administrative controls that meet or exceed industry best practices for data security and patient privacy. Labs often ask us, how will it be possible to recontact patients for research opportunities when their information has been de-identified? Well, it's hard. And that's exactly why in the future, we may want to be able to collect identifiable patient information so that they could be recontacted more easily. Until that happens, researchers may be looking to enroll a specific patient in a clinical trial based on that patient's tokenized profile. However, we will never re-identify a patient based on its de-identified cryptographic hash. Instead, an external IRB approval will be obtained for us as your business associate to contact a broad group of patients based on broad criteria that will not identify one unique patient individually. The trial sponsor may hope that the particular patients of interest choose to enroll in the trial, but we'll cast a broader net using information that's available without re-identifying patients. Now, we've talked a lot already about the importance of patient consent and acknowledgement of research testing. So I want to spend some time talking about the provisions in the network participation agreement that relate to patient consent and authorization. It's important that your lab shares the following principle with us if you're going to join the Ovation Research Network. This principle, even though patient information will be de-identified, patients should understand that they're participating in research and how that participation benefits the world. When it comes to legal responsibility, your lab must attain any required consents or authorizations that are required by all applicable laws to use de-identified patient specimens and information for research purposes. While we cannot assume this responsibility on your behalf, our experts will work closely with your lab to adopt a patient consenting and notification strategy that is both amenable to your current operations and that meets these highest ethical and legal standards. Now, United States federal regulation states that the process of de-identification mitigates privacy risks to individuals and thereby supports secondary use of information for life science research. This means that provider attestation you're receiving today, that is, that they obtain informed consent for the patient to receive the needed medical testing, is legally acceptable as a means of consent for de-identified research, as long as that language does not explicitly preclude research testing. However, we firmly believe that notifying patients about the usage of their remnant specimens in research is the right thing to do. As a result, we have a short and a long-term goal that we'd like your lab to commit to as part of this agreement. These are outlined in the consent overview slide. As I said, our experts will work closely with your lab to ensure the legal requirements for remnant sample use are met, and that there's a notification strategy in place that best serves the needs of you and your patients. That notification strategy is shown as an Exhibit A to the network participation agreement, notated as required consent language, and we will expect over time your lab to adopt this particular notification language so that no patient is ever surprised by the use of their research specimens. Provider attestation that you're already receiving today, again, can suffice. And before you sign our NPA, our legal affairs team will meet with your team to discuss this strategy, exactly what it will look like for your lab, and the details as they depend on your exact client portfolio, your test ordering process, and more. So some common questions about the consent process. Labs ask, will a patient ever need to provide a written signature of consent for secondary research testing? Well, as I mentioned, federal regulations do not require patient consent for research. But as a best practice, we're asking that patients be notified about secondary use of their specimens. This notification can happen in a few ways depending on your current operations. It may be that gaining a written or electronic signature of acknowledgement from the patient works well with your existing flow, but we don't require this. Most commonly, when the specimen is collected, the provider will verbally inform patients that their remnant specimen information may be de-identified and used in research after clinical testing on the specimen has been completed. Then the provider will attest that they notify the patient of this secondary research use when they place your order for the test with your lab. Now, again, this process can vary significantly depending on your client portfolio and test ordering process, which is why you'll have a more detailed discussion with our clinical affairs team before signing this network participation agreement. I'll also add that the Ovation software that we'll provide for you tracks these notifications and research entitlements associated with every specimen and patient data. Makes it easy for you to track this information, makes it possible for us to verify those entitlements before a specimen is ever used for research purposes. So finally, let's talk about how your lab can qualify to perform profitable research testing services and how those services are contracted. One of the core tenets of the Ovation Research Network is to provide diagnostic labs that participate in the network with the opportunity to perform laboratory testing that turns leftover specimens into real-world data available for research. Now, your costs associated with participating in the research network your costs associated with participating in the research network will be covered by reimbursement, but you'll be allowed to receive additional revenue for performing research testing services. The form of network services addendum shown in Exhibit B of the network participation agreement will be completed for each time that you perform research testing services for Ovation. This form may be used for other network services as well, including storage of specimens for the network. So how is it that labs are eligible to perform research testing? Well, your lab's eligibility for this testing is based on, first, your ability to follow our research team's qualification process to receive the research testing business. Of course, we want to make sure that all the data coming out of our research network labs is consistent from a quality level. Second, your lab's capabilities to perform the research testing required based on the research questions of our project. As an example, the ability to perform whole genome sequencing. And finally, your capacity to perform research testing at the time it's needed for a project. When a project is initiated by Ovation and it's determined that we need to perform additional testing on specimens, our preference and the design of the research network is to bring that testing back to the lab that contributed those specimens to the network. We'll reach out to your lab first, and then all the other labs in the network that have capabilities to perform this testing as needed. We're always looking to characterize specimens as fully as possible when we pull them out of our repository, which is why we're typically performing a wide range of testing. We typically do total nucleic acid extraction from these specimens, collecting both DNA and RNA. Then we perform whole genome sequencing, RNA whole exome sequencing, or targeted characterization using quantitative PCR or other methods. In some cases, we're determining disease and normal tissue and doing macro dissections from FFPE tissue blocks. As you can hear, there's a lot of services that are necessary and a lot of testing that's performed in the network, and almost any lab can perform at least some of these research services that we're doing within the network. All right, that brings us to the end of the presentation and the walkthrough of the network participation agreement. I'll pause and see if there are any questions from the group.
Hey, Barry, we did actually get a bunch of questions that came in, but we're already 2 minutes over. I think we went a little longer than expected, so apologies about that. What we'll do is we'll follow up individually with those who asked questions, and we'll aim to do that in the next 24 hours. Sorry about that, but went a little bit over time today.
Great. So what happens next? Well, we've highlighted what we think are the most critical topics, but please be aware this presentation is intended to be used for your general information only. You should, and your team should, review the network participation agreement in full with your own legal counsel. Once you've had a chance to review that document, you'll meet with our clinical affairs team. We'll discuss your specimen types, your current consent procedures, so we can determine how we're going to work together in more detail. We'll answer any of your legal team's questions about the network participation agreement as well in parallel. Ovation will then develop a work order that describes the scope, approach, and details, including timeframes for how our team will work with you to assist in implementing the Ovation Research Network activities in your lab. And that document will be informed by your meeting with our clinical affairs team. To join the Ovation Research Network, you'll sign the network participation agreement and the work order at the same time, and then we'll get started. We're thrilled that you were able to join us today. Thank you for taking some time with us. We really look forward to the opportunity to work with you on the Ovation Research Network and to advance life science research and discovery together. Please reach out to us, your Ovation representative, with any further questions or concerns. And we want to ensure that this agreement is as transparent as possible. We look forward to hearing your thoughts. Thanks for joining us today.